Okay, great. Okay, so this is the Senate Bill 85 meeting of the Commission to Study Environmentally Triggered Chronic Disease. I'm Mindy Mesmer, the chair of the commission, and joined by a few members right now. Hopefully some more will join us as we move into the meeting a little bit further. We have a special presentation today. I took attendance, so I'm tracking attendance separately. But we have a presentation by Dr. Elijah Stommel. I hope I'm saying your name right, Dr.
Yes,
Stommel.
that's correct.
Awesome.
Can you hear me?
We can hear you, but we can't see you though.
Okay, I'm just trying to get this lecture set up properly.
Okay. And I think as give it a try to share it. I think you should be able to share. I just don't see the.
Just give me 1 second. You can do whatever your guys are doing.
Sure. Let's see, Peg, do you want to just give us an update on education to start out while we're getting started today? Do you have anything to talk about?
Well, since the last meeting, I did draft up a letter. Mindy, I think you've seen it. Representative Murphy has seen it, and Dr. Timmons has seen it. And there are a few iterations, but it seems like we're ready to go. This is a letter that will be sent to primarily CEOs, hospital CEOs in the southern tier, where PFAS contamination is, more significant than other parts of the state. And it's an invitation to have a discussion about how we as the education subcommittee can best get information to the clinician community, particularly the physician community, because that's been a group we haven't yet been able to capture. We're covering the nurse practitioner community pretty well. We're set up again in April to present at that conference. So I think that we just need to have a little discussion on specifically who the letter is going to go to, and then we will be all set.
Great. And Peg, can you, once it's finalized, I know there was a little bit of adjustment to it. Once it's finalized, if you would share it with me, and then I'll share it with the commission, because I think the rest of the commission, just to keep people informed about the wonderful things that you guys are doing.
Great. I will do that, Mindy.
Excellent. Any questions about that update from commission members? Great. All right, Dr. Stommel, we're so happy to have you with us. We've been talking about having you come speak with us for quite a while, and we're so happy that you're able to join us today and speak about your important work, I believe in Vermont, but also in New Hampshire. Is that correct, on ALS and cyanobacteria?
Yeah, actually, in some studies, we've looked at the entire country, and others have involved Ohio, which we really studied carefully for a while.
Interesting.
I have you can see the lecture, the first slide.
Yes, but if you could, hit close in the lower left where it says close, if you don't mind.
Oh, okay, let's see.
There's a white button sort of in the left-hand section of the screen.
I have everything on the top now, since I.
Oh. It says document recovery, and at the bottom it says close.
I don't see anything at the bottom. I can unshare it. How do I unshare it?
What you could do instead is, that at the bottom right-hand side of the screen, there should be a little picture that looks like an open box on top of a stand. If you want to just hit that, it would bring it out to the whole presentation.
No, that's not working.
There you go. Yeah.
Is that it?
There we go. That's it. Yep.
Okay. All right, great.
Well, I'll let you take it away. Introduce yourself if you want. Just do a short introduction for the commission, and then take it from there.
Yeah. So I'm a neurologist at Dartmouth Hitchcock, and I've been studying risk factors for ALS for many years now. And the whole issue of cyanobacteria has been very interesting and worrisome throughout northern New England, where I've spent most of my time looking. But elsewhere as well. So I have a big team of people I work with. It's a multidisciplinary type of research, and we have everybody from epidemiologists to clinicians like myself to geographers who use GIS mapping techniques. We have statisticians. We've been looking at heavy metals, nanoparticles from air pollution, neuropathologists, and geneticists. So it's a big team, and even remote sensing experts who use satellites to visualize cyanobacteria on the ground from up in space. I have no disclosures. I do have funding from various agencies. So I don't know. I assume that most of you have known somebody with ALS in your lifetime. It's considered to be a rare disease, but it's actually fairly common in my world. I see about 50 to 60 new cases of ALS a year, and I'm seeing on average 150 plus patients at various stages of their disease when they come back to clinic. It's a devastating disease. It generally spreads fairly rapidly. The lifespan's about 2 to 5 years from the time of diagnosis. It involves both upper and lower motor neurons, so it causes spasticity as well as weakness and atrophy. Patients generally die from respiratory arrest. They lose their autonomy. They often will be unable to walk or move their arms or legs and need somebody to take care of them around the clock. Some of them are on ventilators. There's clearly genetic influences in getting the disease, but there's also environmental influences, which is my biggest interest. I've seen a few patients that have survived for prolonged periods of time, but it's generally a fatal disease. The famous physicist in England, Hawkins, had it for 35 years. But of course, people who have more resources and can get round clock nursing and have it all paid for, tend to do better. I've had a few cases where it actually arrested and the patients were fine with some deficit, but continued to survive and function on some level. It's a prion-like disease, so it starts in one part of the brain or the spinal cord and then spreads. So it might involve one hand first and then move to the other hand and then into the bulbar muscles in the throat. And it characteristically spares some systems like movement of the eyes, and bowel and bladder function is generally retained. It's devastating to families, of course, as well as patients. As I said, the patients lose their autonomy. There really aren't any good therapies. And there aren't a lot of registries out there to study the disease. And I'll mention that a little bit more later. The financial burden, the average patient will have their insurance and themselves will have spent about 1.5 million by the time they die, and about 9% of that is paid by families, 6% by charities. So, and of course, if you're a breadwinner for the family and you can't work anymore, it has a huge financial impact there. And of course, the average onset of disease is somewhere between 55 and 65 years rather. And these people are still working and trying to get their kids through college or whatever. It's a devastating situation. And that statistic is 10 years old. So I'm sure it's more expensive now. I don't think I'll go into the various pathologies, but there are many things that can go wrong inside the motor neurons. And what this means is not just one disease. It's many, many diseases. Everybody's genetics is a little different. What's going wrong in one person's motor neurons is different than another, which is part of why it's such a difficult disease to treat. So most people feel that ALS is a combination of genetic susceptibility and exposures in the environment. Parkinson's disease, which is related to ALS in that it's a neurodegenerative disease, by some experts is thought to be totally cured if we could get rid of some of the toxins in the environment, and probably didn't exist at one time. And about 90% of ALS is sporadic, so there isn't any obvious familial aspect to it. You can't trace it back to somebody else in a prior generation. But about 10% is familial Mendelian ALS. And there's a big family in St. Johnsbury, Vermont, called the Farr family, and they have a SOD1 mutation, and it's 50% of the family that ends up getting it. However, some people in those families get it in their 20s, and other people get it in their 80s, and some people don't get it at all. So there's clearly some other something else going on, which is most likely risk factors, environmental factors. Okay, so the genetic component in sporadic ALS, which is 90% of ALS, is relatively small compared to familial ALS, which is about 10% of ALS. And these are some of the typical risk factors that are thought to exist. Familial history is very important. And in fact, even if you have sporadic ALS, it increases your chances of getting ALS in future generations by about 8 times. So there still is some genetic component. It just hasn't been discovered as of yet. There are some genes that have been discovered, but by and large, it's still a mystery. Heavy metals like lead, mercury, cadmium, manganese, arsenic may be risk factors. Welders tend to have a higher rate of ALS, and welding, you're exposed to a lot of manganese when you're melting down metals. Cyanobacterial toxins, I'll talk about. I won't go into all of these. Electrocution, we've done questionnaire data, and it looks like people who've had a bad electrical shock, it might go back as much as 40 years, but still may be a risk factor. Formaldehyde, undertakers have a higher rate of ALS, and there are other metabolites that look like formaldehyde chemically. Smoking may be a risk factor. Athleticism. Most of our ALS patients are very lean and have a history of partaking in all kinds of sports. Muhammad Ali had Parkinson's disease, multiple head traumas. Head trauma is probably a risk factor. Italian soccer players were shown to have a higher rate of ALS. And of course, they're butting their heads all the time. Lou Gehrig probably was exposed to head trauma. The military has about a 2-fold higher rate of ALS. And from the time of deployment, it's about a 10-year lag period before they start getting it. And they start getting it at a younger age because military recruits tend to be younger. What they're exposed to in the military includes all kinds of bad things like exhaust fumes from jets and trucks, all kinds of organic solvents that are used to clean motors. And they get all kinds of vaccines, and they get the vaccines with a pneumatic gun usually, which is quite traumatic. In the Iraq War, they were exposed to burn pits, dioxins, maybe head trauma, and many heavy metals. They're shooting guns, they're exposed to lead. Let's see, pesticides, very strongly linked to Parkinson's, not so much with ALS, but there probably still is a risk factor there. And then persistent organic pollutants, things like fire retardants, which are sprayed out of airplanes onto forest fires. They're not only carcinogenic, but they probably will cause neurodegenerative diseases, including Parkinson's, ALS, and maybe even Alzheimer's disease. If you take 2 identical twins, and they usually get separated at about the age of 18, where one goes off to college maybe, and the other one goes into the military, or takes a job, doing something. Their diets change, their habits change, but they have exactly the same genetic makeup. What do you think the risk of if one identical twin gets ALS, what do you think the chances of the other identical twin getting ALS is? Can somebody guess? I can't hear you. Can you hear me?
Yes, we can hear you. Over 50%.
It's about 25%.
Oh, really?
So it's really rather, you would expect it to be, close to 100% if it were all genetic, right? But it's not. And I don't have time to go into epigenetics, but some environmental factors will turn off a gene or turn on a gene. And if you look at a chromosome in a 3-year-old identical twins, they look pretty much the same. When you get into your older years beyond 50, the genes that are turned on or off are quite different. And, people look different as they get older, but if you would expect identical twins who had exactly the same diet, exactly the same good habits, exactly the same bad habits, or exactly the same jobs to look the same, but they don't. And these are just some examples of that. Back in the 1950s, there was this theory called the Armitage-Dole theory of cancer. And they looked at huge databases of cancer patients, GI cancers for the most part. And they showed that there was a logometric slope of about 5 when you plotted the age of onset versus the incidence of disease. And this slope of 5, the theory is that you would need 6 hits over time to develop one of these GI cancers. So if you got exposed to A, B, C, D, E, F over a period of time, you might develop a GI cancer. The first step might be genetics, and then everything else beyond that would be environmental factors. And GI cancers probably are very dependent on what you eat. There was a big database in Europe where they did exactly the same plot, and they showed ALS had exactly the same plot as the GI cancers. So it suggests that there is, over time, a series of hits that you're exposed to, and at some point, it breaks the camel's back and you end up getting the disease. In ALS, there are many things that can go wrong inside your cells, inside your motor neurons. Some of that is RNA metabolism, protein, the handling of proteins inside of cells. The cytoskeleton, which is the skeleton that makes up the cells, can be perturbed. And things like pesticides can affect protein homeostasis. The epigenetics, which I talked about in the previous slide with the twins, can affect various elements inside the cell where things can go wrong. So there's this interaction between environmental things and the structure, the protein handling of the cells, and the RNA metabolism. Those are the big ones. But there's also things like neuroinflammation and something called apoptosis, which is programmed cell death. There are all these different things that are involved. Mitochondrial function. So when do these exposures happen in ALS? In the military, as I just told you, people will be deployed in the military, and then 10 years later, they'll start to develop ALS. I'm going to talk about Guam, which was a sort of a Pandora's box for epidemiologists back in the 1940s, where there was a 200-fold higher rate of ALS. And people were moving there from the Philippines, and other people were moving away from there to, say, Los Angeles. And they still had the risk of developing ALS, even though they were 30 years from the time either they moved to Guam or the time they moved away from Guam. So something in the environment takes a while to cause the disease. And it's been postulated that even in utero or in infancy, one of those first insults may occur. This is an interesting study done by a colleague of mine, Manish Arora, who's in New York City, actually trained as a dentist, but he looked at children's teeth, and he was able to show that there were copper and zinc metabolism abnormalities in the teeth of children. So he can take those teeth just like the rings of a tree, and he can drill a little hole with a laser and measure what metals are there at various stages in utero. And he was able to show that there was this zinc and copper defect. I'm sorry, zinc and manganese defect, even in utero. So that is linked to autism. So it could be that's happening in ALS as well. So in Guam, back in the 1940s, when the Americans took away Guam from the Japanese, the people there were treated very badly by the Japanese. They were basically in a starvation mode. And on the left, you can see a woman with ALS who's got atrophy. She can't stand up anymore. She can't move her arms. In the middle, there's a man there who's got a blank stare in his face, and he has Parkinson's disease. And then there was also a dementia-like complex. So this was called Litigo-Bodick disease. The rate of ALS is about 200-fold higher than the rest of the world. So it wasn't Pandora's box, it was the Rosetta Stone that I was talking about. A lot of epidemiologists moved there and tried to figure out why there was this higher rate of ALS and Parkinson's on this isolated island in the middle of the Pacific Ocean. There's a great book written by Oliver Sacks about this, and it's worth reading if you haven't read it. So they were sort of in a starvation mode, the people who lived there, and they were eating the flour from cycad seeds, which you can see on this picture in the middle. You can see those seeds on what looks like a palm tree, but it's a cycad plant. And the natives there knew that they were sort of toxic because they didn't wash this flour that they made tortillas and things out of. It would cause GI upset and burning in the mouth and some other symptoms. So they had to wash this flour over and over again. And so some of the early toxicologists and epidemiologists that went there started studying this flour, which was sort of the mainstay of their diet. And they found an amino acid in the flour called BMAA. And they found some other toxins in there too, but I won't go into those right now. The BMAA comes from a cyanobacteria that lives in the roots of the plant, of the cycad plant. And on the upper left there, you can see what's called coloroid roots. So they look like coral, but when you cut them in cross section, they're green in the lower left. And that green is cyanobacteria. And in the upper right, there's a flying fox there. And in the image just to the left of that is a flying fox upside down eating a cycad seed. The flying foxes were a very popular food in Guam, but they weren't able to catch them readily. And so when the Americans took over the island, of course, the natives got a hold of guns and started shooting the flying foxes and eating them. They eat the hair, they eat the bones, they eat the whole animal. And a friend of mine, Paul Cox, went to the British Museum in London and got some samples from some of these flying foxes and found them to have very high levels of BMAA, the neurotoxin that's produced by the cycad, the cyanobacteria in the cycad plant and gets into the flour. Another big discovery was that when the BMAA was first discovered in the flour, they were missing about 99.9% of the BMAA because it was protein bound. So when chemists later on started to study this more carefully, they found that the BMAA was much more abundant than was previously thought to be. Initially, they thought you would have to eat your weight in cycad flour every day to get a neurotoxic effect. The natives had basically annihilated the flying foxes in Guam because they shot them all. And an insect was introduced from China, and almost all the cycad plants are gone in Guam now. The rate of ALS in Guam has gone down to what it is in the rest of the world, but there still is a fairly high rate of Parkinson's disease and a dementia-like disease. So BMAA is the structure in the upper right here, and it's associated with ALS. And there's another disease that causes a spastic paraparesis called lathyrism, caused by an amino acid called BOAA, which comes from chickpeas, and it's also in the starvation mode. So in the Far East, there are certain chickpeas that will produce this BOAA, and when they eat too much of it, they'll get lathyrism, and they'll get wasting and spasticity in their lower extremities. L-serine is an amino acid that looks somewhat like BOAA and BMAA, and that will actually block the effects of BMAA in a monkey model, which I'll show you next. And it's thought that the BMAA gets into the translational process where mRNA makes proteins. And when it gets into the proteins, it makes them misfold. And misfolded proteins, of course, is one of the hallmarks of neurodegeneration. So in New Hampshire, back in 2008 or 2009, I don't remember the exact year, I had students, of course, who want letters of recommendation so they can get into medical school. These were undergraduate students from Dartmouth. And I had them map out all our ALS patients that we had in our data banks. And lo and behold, we found clusters around ALS around some water bodies. And this in particular is Lake Muskoma in Enfield. Since this map was made in 2010 or 2011, we found about another 15 patients that are around this lake. And I had no idea why there would be so many patients around this lake. It turns out that there is a high mercury content in the sediment from Lake Muskoma because it used to have a lot of tanneries around it, and mercury doesn't go away in any quick way. But I was talking to a lake biologist from UNH, Jim Haney, and he said, "Well, Lake Muskoma has terrible cyanobacterial blooms just about every year." And so I started looking at that. And of course, one of the first things that came up was the Guamanian story on the island of Guam. And it turns out the BMAA is quite plentiful in the fish from Lake Muskoma. This is a carp, about a 20-pound carp that came from the Shakers ponds that they had back in the 1938 hurricane. The Shakers were eating these carp, and they had lakes, little ponds rather, around their village there. And the carp all got washed out into the lake back in 1938. And some of the carp in Lake Muskoma get up to 100 pounds. And they date back to the 38 hurricane. Anyway, we were able to find BMAA in the carp there. We've also been able to find it in aerosol. So we've set up aerosol collectors around the lake, and we've been able to find BMAA in its neurotoxic isomers there. We did some questionnaire data, probably now up to about 700 ALS patients. And we found that water skiing and some water-related sports like windsurfing is a risk factor for ALS. And there's one publication there that we did in 2017. There is a study by Backer et al. Where they did nasal swabs and also set up aerosol collectors around lakes that had cyanobacterial blooms in them. And they've been able to find one of the cyanobacterial toxins called microcystin in the nasal cavities of people that have been water skiing behind boats in these lakes in California. Microcystin is thought to be a liver toxin, at least historically, but it also is a neurotoxin. And cyanobacteria make over 100 toxins that are toxic to humans, but there's at least 5 or 6 that are neurotoxic and will cause damage to neurons. Here's an advisory from the internet from the DES in New Hampshire. And you can follow where all the blooms are in New Hampshire over time. In Lake Muskoma, this is Anabina. This is a microscopic image of cyanobacteria. And these are the same exact cyanobacteria that are found in the cycad plant in Guam. Here's an eastern shore of Lake Champlain. In the middle, you can see the cyanobacterial bloom there. If a dog were to go down to this bloom, and they often do, and try to eat some of this green matting, it will kill a dog in about 3 to 5 minutes. And there's a lot of domestic animals that are killed by cyanobacterial blooms if the toxins are ripe and they're there. These green dots on the left are ALS patients, and these are bona fide clusters of ALS along Lake Champlain in Vermont. And you can see how close some of them are to Lake Champlain, which has terrible blooms in the summer and even into the fall and early spring. If you think about it, what are the prevailing winds in New England in general? They're from the west or southwest. So it's interesting to think that aerosol may well be important here. Here's a blow up of patients near or on Lake Champlain. This is an interesting thing. I have what are called conjugal couples, or people that either habitate the same building or are married and live in the same house. In this case on the left, I had a patient, very wealthy patient, who lived on Shelburne Point, and he moved after he got divorced to a less palatial mansion. But I didn't know that originally. And I diagnosed him with ALS around, I think it was about 2014. I don't remember the exact year, but a couple of years later, I diagnosed his caretaker, who took care of his estate out in Shelburne Point. And so they were basically living and working in the same environment. And the Shelburne Bay area gets bad cyanobacterial blooms in the summer. And this guy had a power boat. Of course, he was out on the lake a lot. This is a small pond on the right near Tilton, New Hampshire, yeah. And there was a couple. The wife got ALS first. She described taking a rake to get the cyanobacterial matting away from the shoreline so her kids could go swimming in the summertime. And about 2 years later, her husband came down with a form of ALS. It's pretty compelling. It's not proof, but it's compelling. Unfortunately, I didn't get autopsy samples from any of these patients. There is a movie that was made in part about the study on the left, and I think it's called The Lake Effect, and you can see it on public television. I think occasionally they have it on there. We set up some filters, aerosol collectors in many lakes that were affected by cyanobacteria. And to make a long story short, you can find BMAA and if not BMAA, some of the isomers, which are equally toxic in many of these water bodies that we checked. So I'm just going to reiterate how important it is that registries be set up for ALS. Some of you may know that I've tried to do that in New Hampshire. We got very close in New Hampshire, and we got the cost down substantially, and I was going to take responsibility for the cost, but then Dartmouth Hitchcock didn't want to be responsible for any kind of a budget. So they basically didn't want to pursue it. Massachusetts has had a reportable registry dating back to 2008. And it was set up because the governor of Massachusetts, Governor Cellucci, came down with ALS, and they wanted to do something in his honor. So that's been the only registry until recently. We've set one up in Vermont and Maine in the last 2 years, and we're starting to collect data for them. And kudos go out to Vermont and Maine for setting them up. Was a monumental effort. There was a lot of pushback, but I think it's going to be helpful. And when you look at, those maps that I showed you, there may be patients that aren't in those maps that should be, and there could be, although all the patients that are on those maps are for real patients. In some cases, there are patients that are misdiagnosed, and registries are very important. For not only for that, but for giving patients information so that they can hook up with specialty clinics, ALS clinics of excellence where they can get good care. Many reasons to have registries, and they've been set up for cancer and many infectious diseases for decades and decades. And I think they should have them for some of the rare diseases as well. We're still hopeful that some of the big states like Michigan, Florida, Texas, California will eventually come on board. That would be very helpful. There is a national registry, but it's not reportable. So meaning it's not mandatory. So even there, it's helpful, but it's not a reportable registry. There was a study done on vervet monkeys in St. Kitts, where they were fed BMAA. Some were fed BMAA in bananas, some were just fed bananas as controls, and then some were fed L-serine, which is the amino acid that BMAA substitutes for. And they were able to reproduce the pathology of Alzheimer's disease and Guamanian ALS in these monkeys. The monkeys didn't actually come down with disease, but they were sacrificed and the pathology was there. And I did EMGs on these monkeys that have large teeth. So I was trying to see if they had evidence of ALS and wasn't able to demonstrate that clearly, but the pathology is clear. So this is sort of a tauopathy, which is what you see in Alzheimer's disease. And this is some of the tangles you see, neurofibrillary tangles you see with Alzheimer's disease. Tauopathies are also very common in the ALS in Guam. And then you can see that the control group, hopefully you can see, the control group, which didn't get BMAA, had the lesser amounts of the neuropathology. And the L-serine group that got L-serine showed much less statistically significant, much less neuropathology consistent with Guamanian tauopathy, Guamanian ALS, and Alzheimer's disease. We tried to do a trial in L-serine, and we've had issues with it. I can't go into those. But we have been able to show a statistically significant reduction in ALS progression in those who get L-serine. So some of the other neurotoxins that cyanobacteria produce, one of them is very interesting, and I don't have time to go into all of them, But antitoxin AS works the same way organophosphate pesticides work. It's an acetylcholinesterase inhibitor. And as I said, organophosphate pesticides are very highly linked to Parkinson's disease, but also to some degree to ALS. And the typical pesticides that are put on farm fields are organophosphate pesticides. Antitoxin AS is 1,000 times more potent than those pesticides put on farm fields, 1,000 times more potent. And it's one of the toxins that will kill a dog in about 5 minutes of ingesting the cyanobacterial mats that are on the shore of many lakes and ponds. Microcystin is a toxin. I guess I have a slide that I can tell you more about. Saxitoxin is a sodium channel blocker, very, very neurotoxic. And cylindrospermopsin is very toxic and works on the cytoskeleton. Here we have a very unusual cyanobacterial toxin called Atochthonus. I can't pronounce it properly, but it lives in the milfoil in certain lakes, especially down in the south of this country. And the coots go down, and the coots are a type of duck. They go down and they eat the milfoil and then inadvertently eat this form of cyanobacteria. And the cyanobacteria causes a myelinopathy in the spinal cords of these ducks. And the ducks become disabled. And of course, then the eagles come down and eat the ducks. And the eagles develop the same disease. And it's wiped out quite a bit of the coot population and a good part of the bald eagle population in the south. So it's not exactly the same process, but it comes from cyanobacteria. We have ways of mapping out the cyanobacteria. In this case, we can look at concentrations of cyanobacteria along Lake Champlain. We have remote sensing data, and we're able to tell where the cyanobacteria are most prevalent. And my friend and colleague, Nate Torbick, who works on the seacoast of New Hampshire, has got NIH funding in the past, and he's now mapped out the entire world. And he can look back over time, about 30 years, and see where the cyanobacterial blooms have been over time. And we can use LexisNexis data, which is credit card data, and tell where patients have lived over time over about 30 years. So we can, with all these fancy artificial intelligence techniques and remote sensing techniques, we can actually see correlations. Here's a map of cyanobacteria in Ohio, which we studied quite carefully. This data is kind of old, but Nate Torbick did remote sensing in northern New England, where we had a fairly robust population of ALS patients. And he was able to show statistically that where the blooms were, there was a statistical correlation with where the ALS patients were. And in fact, it looks like with mathematical modeling, those ALS patients tended to be to the east of lakes that had cyanobacterial blooms. And again, the prevailing winds are from the southwest. So BMAA may well represent one of these 6 hits that I talked about earlier in this lecture, and may be one of the things that are in the environment that interact with the genetic makeup of patients. And the same may be true in diseases like Alzheimer's and Parkinson's disease. So microcystin, which I talked about, is very prevalent in cyanobacterial blooms. It's been shown to break down the blood-brain barrier, which is very important in protecting the brain from toxins. And so it's actually, and it also causes inflammation. So this is some other work that Nate Torbick did back in 2018. And we took our ALS patients and we're able to show where they were in relationship with cyanobacterial blooms. We've since been interested in looking at aerosol, both in terms of filter collection, but also in terms of lung tissue and bronchial alveolar lavage samples. So people go to the clinic and they go to the pulmonary clinic and they get bronchial endoscopies done. And we were able to collect fluid samples from these all takers just going into these labs. And we've been able to detect cyanobacteria in both postmortem and in lung tissue from bronchial lavage. We can use fancy probes with PCR which is the same technique that's used, to look for genetic makeup in homicide cases, that kind of thing. Anyway, we can find cyanobacteria in the lungs, and here there's a detection limit here where the horizontal bar goes across the dotted bar, and a good deal of our postmortem cases do have detectable levels of cyanobacteria in their lungs. And what we'd like to do is to correlate these with the neuropathology of the same patients, and we're in the process of doing that. We don't have enough data yet. This is the bronchial lavage data where we actually did publish this back in 2018. We're able to find cyanobacteria both in the nasal cavities and in the lungs of patients. And we weren't able to correlate what goes on in the nasal swabs in the nasal cavities with what goes on in the fluids in the lung, but that may have to do with the physiology of the lung. And we've also been able with with mass spec and with ELISA assays to be able to find some of these toxins in the lungs of the same patients that I just showed you. And here's some samples. We can find BMAA, we can find microcystin, we can find M-toxin A, cylindrospermopsin, all these different neurotoxins that come from the cyanobacteria. This was a recent study where in Alzheimer's patients, this group looked for BMAA, was able to find it in the olfactory tracts, in olfactory tracts where the nasal cavity ends, where your sense of smell comes from. And we're able to show the neuropathology of Alzheimer's disease in the olfactory tracts and in a dose-dependent fashion with the amount of BMAA they found there. So the more BMAA, the more neuropathology was found. And they were able to track this right into the brain itself and show the neuropathology in the parts of the brain where the olfactory tracts go to. This is just, you can see the olfactory tracts. This is the bottom of the brain, and you can see the two snake-like structures in the dotted box there. Those are the olfactory bulb and olfactory tract, and that's what they were looking at. And it's well known that Parkinson's disease and Alzheimer's and ALS all have smell deficits as their disease progresses. And actually, one of the first symptoms in Parkinson's disease is a lack of smell. And we use that in the clinic. So, are these aerosolized toxins, be it cyanotoxins, or be it air pollution, or formaldehyde, or any number of things, are they getting into the brain through the olfactory tract? Or are toxins getting into the brain through the GI tract, or through the skin even? these are all being studied. This is just showing the neuropathology in the study that I just described, where they can show that all the neuropathologies of Alzheimer's disease in a BMA concentration dependent manner. So long-term exposures are important, and that's something we've been looking at. And we use this credit card base, so we can take a patient, get their name and their social security number, and then this company can give us where every patient has lived over 30 years. And then we can put all the toxins on a map, a geospatial map, and we can see what people have been exposed to over 30 years, 25, 30 years. We can do the same thing with the algal blooms. There's a big popularity in spirulina, which is a supplement that comes in all different forms, quite popular in young people. I was just contacted by an ALS expert in the state of Pennsylvania, and he's seeing a lot of young people coming down with ALS. And with his questionnaire data, he's seeing that many of them are eating spirulina. And spirulina does contain BMAA. That's been shown in several studies. And there's a guy in the middle here who's sort of collecting the cyanobacterial gnats in a pond where they're manufacturing spirulina. So that's worrisome. Maybe there are other ways that we're ingesting it. Fish, some fish have high levels of BMAA. Our lab is not just studying BMAA, we're looking at other things, of course. And this is just to give you an example. This is a nationwide map of pesticides. In this case, the herbicide glyphosate Roundup. We've shown that it seems to be associated with ALS. And then there are other pesticides and herbicides that seem to be statistically worrisome for ALS. I don't have time to go into it in detail. And then there are some heavy metals and that are in the upper right. Those are the statistically significant ones that may be important. And then there are airborne volatile toxins like styrene and TCE, which is used in dry cleaning, but also is used in a lot of manufacturing that may be associated with ALS. So there are many different hits, but we feel that cyanobacteria may be one of them. So I'll stop there and answer any questions if you have them.
Thank you so much for that presentation, Dr. Stommel. It's comprehensive, but take questions. I see Senator Riccardi has her hand up right now from commission members.
Thank you. First of all, thank you for such a really thorough and very informative presentation. I have a couple of questions. Sadly, I lost my uncle this week, and I'm mentioning it because he all of a sudden, it seemed like they said he had ALS, and then it turned out it's also Parkinson's disease. And I mention it because my aunt told me that she believed, because he was such an avid golfer and 3 of his best friends also died from the exact same thing. They all golfed together, but they didn't live in the same neighborhoods or anything. And after listening to you, it sounds like that might be plausible.
Absolutely. And there are some anecdotal stories and publications where golfers do seem to have a higher risk of ALS. And I had a colleague named Robert Brown, who's actually a famous ALS researcher from UMass. He used to be at Mass General. And he told me the anecdote that golfers often lick the tee for good luck. And if you've ever looked at the ponds on golf courses, they're bright green.
Yes.
And if you've ever seen how immaculate golf courses are, of course, they're all sprayed with herbicides and pesticides. yes, I think it's quite likely. And that also could explain why soccer players or why football players seem to have in part, a higher rate of ALS. It's not only the head trauma, it could be other things that are on the turf. I
And just
tried to get data on Lou Gehrig, going back to Yankee Stadium in the 1930s. I thought maybe a historian there might be able to get me some information on the groundkeeping there back in the 1930s, but I haven't had any luck so far.
And then one follow-up, if I may. You also spoke about the nasal passages and the blood-brain barrier, but of course, those were things that were checked after passing. Is there, or are there tests now that people can get that are accurate to check the blood-brain barrier or the nasal cavities, or whether they have neurotoxins, toxins, or heavy metals in their system?
Well, there are ELISA assays that you can purchase, but they probably need to be done in a laboratory where you can check for things like microcystin. They are commercially available. There is one for BMAA, but it's of questionable validity. We did nasal swabs on patients, all comers that were going to the pulmonary lab, and we were able to find cyanobacteria in the nasal swabs. So people are breathing them in. And, if you're a runner and you're running along Charles River, or around Lake Muskoma, or along the Lake Champlain, you're almost certainly going to breathe in some of these bacteria that are being aerosolized. And so, yes, I think they're probably in abundance, especially when the air conditions are right. When you have fog on a lake, when the air temperature is cold and the lake is hot, or the other way around, the amount of bacterial debris in that mist must be incredibly high.
Thank you so much.
And don't forget farm fields. A lot of pesticides put on farm fields. And if you see somebody plowing a field when it's really dry, you see that aerosol behind the tractor. I have seen many veterinarians with ALS. And I suspect that they're not only are exposed to animal viruses, but they're also exposed to pesticides, and they're also exposed to cyanobacteria. So, there's a lot of very interesting observations that haven't been fully understood at this point.
Any other questions from commission members? I see Dr. Timmons has joined us. I have a bunch of questions if others don't. That was an interesting thing about veterinarians. Do you think it's from touching the animals maybe that have pesticides on their fur,
or they're working on large animals and they're out in the farm fields day in, day out. And, of course, farm field ponds, if you've ever seen those, they're bright green too.
Yeah.
And I know where I live in Orford. I don't live in Orford, but I go up to Orford, New Hampshire sometimes on the River Road, and there's some incredibly green ponds up there. I mean, they look like a jewel,
It's very bright green.
Yeah. And
I have a couple of questions. Oh, go ahead. Sorry.
If you are in an airplane and you go through the Midwest, or you come into Logan Airport from the south, you'll see farm ponds all over the Midwest in the middle of agricultural areas that you can see are green from an airplane. And the cranberry bogs coming in from the south towards Logan Airport are often bright green in the summertime. So I'm sorry, go ahead.
No, go ahead. Anyone else on the commission with questions? I have a bunch I'll just go into if nobody has any. Rosalie, do you have a question?
Yes, I do. Thank you for the presentation. So, there is a very high prevalence of this in the Midwest, like Lake Erie, Michigan, I'm sure, where there's also cyanobacteria blooms in that lake. What is it? Well, let's just stay with the state of New Hampshire. What is the state of New Hampshire doing to protect the public when there is a cyanobacteria bloom?
They put up signs.
Mhm.
They have a Department of Environmental Services. There's a website, and they put, but people walk right by the signs, and
Yeah.
and they have a picnic and then go swimming, and you can drag a horse to water, but you can't make them drink necessarily. And I think registries are important. I think public awareness is very important. They'll often in Vermont, when Lake Champlain heats up in the summertime, it'll be on the television in the evening if you watch the local news. I heard recently that the prevalence of ALS in Vermont may be much higher than we think, but that hasn't been published. But I've heard rumors to that effect.
Is there a length of exposure? If you go to that lake and recreate one day and you inhale all the spores, is there any association with, do you have to have have you found in your research, the length, does the length of exposure correlate with a higher risk, or?
That has not been figured out. But when we do the migratory history of people, we do get different results than when we just look at where somebody lived when they died. And most of the people on Lake Muskoma that we studied had lived there for at least 10 years. I think all of them had lived there for at least 10 years. And the people who are in the military, they're generally deployed for a year, 2 years, and then they're not in that environment anymore. But it takes about 10 years to come down with ALS. So in the military, so that's the beginning of it anyway. So I can't tell you, what the dose you need, and everybody's different, and it may be partly what else you were exposed to. Did you work in a funeral home and were you exposed to formaldehyde and then lived on a lake? Or did you had head trauma when you were a child, or were you electrocuted, or did you make lead sinkers in your basement for fishing? So there's many things that probably play into it.
Yeah. Thank you.
Yeah.
I should have mentioned that's Dr. Rosalie Lepresto. Any other comments or questions from our commission members before I ask mine? All right, so I have a question about Dr. Torbeck's cyanobacteria mapping, and wondered if you said he's able to do it for quite a long time frame. Is there any information available to you about whether those cyanobacteria blooms are getting more prevalent in New Hampshire and New England over the last 25 to 30 years?
They're more prevalent in general, not just in New Hampshire, because of global warming. And
that's what I was wondering about.
Yeah.
Because the warming temperature makes, yeah.
There's a researcher at Duke, I think he is, although his lab is on the seacoast. His name is Hans Perl, P-A-E-R-L, I believe. And he's done some work on that. And Australia has a huge problem with cyanobacterial blooms. And according to their records, the incidence of ALS has doubled there. in the last 20 years.
Hmm.
So
That was going to be my next question. Do you have a feel for, I know we don't have a registry, unfortunately, but do you have a feel for the long-term, the shape of the curve for prevalence of ALS in New England?
I don't want to conjecture on that. I think I need to ascertain my cases a little better, and we should have data on that in the not too distant future. And I do have a grant through the NIH to study the Massachusetts population, and certainly that's something that we can try to answer. It's a good question, though.
Okay. And then I had a question, there was just that recent study that came out over the last few weeks with the younging of cancer cases. And if we're seeing an increasing prevalence of cyanobacteria blooms and we can connect that to ALS, are we seeing a similar younging of ALS onset cases?
Yeah, so I don't know the answer to that one. I certainly see a lot of young people with ALS. Some of the cyanotoxins are actually carcinogenic, so that's interesting. Could it be one of the factors that is involved in increasing rates of GI cancers? That's total postulation. There was a study that looked at the population in the United States in association with liver cancer, which is linked to microcystin, one of the toxins. And they were able to show some statistical significant correlation. So I don't have an answer for that. But all these things, I mean, air pollution is really bad. And if you go to a place like Mexico City, one of the worst places for air pollution in the world, I have a neuropathology colleague and friend who goes there every year and studies the population there, and she does electron microscopy, and she can find the nanoparticles from air pollution, largely from diesel trucks and gasoline powered cars. She can find the nanoparticles in the brains of young patients. These are people who are teenagers, and she can see the neuropathology of ALS, Parkinson's, and Alzheimer's in teenagers. And she's looking at other things like, are these teenagers getting demented there at an early age? Are they getting ALS at an early age? But the neuropathology is there for those three diseases, even in neonates, even in babies in some cases. So it's scary.
I kind of wondered because you said, particularly, you referenced dogs a few times and how it's very toxic to dogs. Some of the
they're stupid enough
toxins.
to eat. They're stupid enough to go
Yeah,
down to the lakefront and eat the mats that taste good to them. I wouldn't think they taste very good, but dogs will eat most anything.
The kids swimming in the water, like that story about the woman brushing away the mats so her kids could swim. That made me think that maybe there was a younging association of exposures increasing in younging children.
Yeah, well, hopefully, I mean the financial impact of cyanobacteria blooms in Vermont is horrendous along Lake Champlain. People can't rent their cabins anymore. And people don't want to go swimming there anymore because the water quality has gotten so poor in some places. And so I'm not sure that, a direct contact is in these really badly affected areas is common. But certainly kids will come home and they'll have GI distress from swallowing water, and they'll get rashes. Whether the chronic effects are going to show up right away, you don't know. And I didn't have time to go into what's fueling these cyanobacterial blooms other than climate change, but asphalt, water runoff. There's no longer buffers between farm fields and water bodies. People fertilize their lawns, and that's why, in part, why golf courses have such green ponds, because they're feeding the cyanobacteria with phosphate and nitrogen.
Did you have that kind of gets what you said a few minutes ago to my other questions. Do you have any feel for, so we think maybe inhalation is one pathway. We think maybe eating fish possibly with high levels of any of the toxins is a possible route for exposure. Is there any other, is it in the groundwater or the drinking water wells? Do you have any feel for what the relative contribution might be?
If you have porous soil, like on Cape Cod, where there's a lot of sand, or many places, some of the toxins can leach into the artesian wells. That's been shown. So yes, you might have to worry about that in certain circumstances, especially if you're near a water body and you have an artesian well. About 4,000 people, the last time I looked, were getting their water directly out of Lake Champlain by throwing the hose off their cottage or their cabin into the lake. And of course, if you take a shower, you're aerosolizing water, and it could have toxins in it. There's many ways you could get exposed.
Okay. My last question was around just the mention you made of L-serine, and wondered if you understood the method of action, why it works, and if it's being used as a prophylactic, especially in areas like the South where we already know, there's also exposure prevalent there.
Yeah, well, if I lived in Florida on a lagoon that had these terrible blooms, or if I lived right on Lake Mascoma, I would probably take L-serine. As I said, it is the amino acid that BMAA substitutes for in the translational process, the production of proteins. So if you flood the system with L-serine, then you might be able to prevent the BMAA from getting into the proteins and causing the misfolding. And that's been shown in cell models and in the vervet monkeys.
No human models, though.
Well, you can't ethically be giving patients BMAA. You certainly can give them
no, but I mean, people that have ALS possibly.
Yeah, I recommend it to all my ALS patients. So I think the vervet monkey study is pretty compelling, and L-serine can cause a little GI upset, but otherwise is well tolerated. And you can buy it in bulk online. They'll ship it to you. Mix it. It's sort of sweet tasting. You can mix it in tea or juice. Some people even just put it in water. So
my last question then is, so from, that you've mentioned the registry quite a bit, what other things that you think need to be done in New Hampshire to address this issue? Just give you an open-ended question.
Well, I think you alluded to having better knowledge for the public, to educate them better, and to maybe put things on the internet and the TV more. That's important. I've given countless lectures to lake groups, to lay groups about this research, and I would be happy to do it on any level that you see fit. I don't know if you have meetings where you have a lot of general public there. I'm happy to give another lecture at any time. It's I think putting some money into it would help. You have New Hampshire Public Radio, you got Vermont Public Radio, I think a good part of the public listened to them, and that would be one venue. State of New Hampshire has a website. There's also the Medical Society. There are ways to get the word out there.
In terms of policy or other things, this commission is a state commission that looks at sort of systematic changes that need to be made to address levels of chronic disease in New Hampshire. So other than the registry, are there other recommendations you can make? And education, of course, is a really good one. Is there anything else that you can think of? I think we're going to discuss internally about whether, and I think we probably should have a presentation from DES directly on this issue about what they're doing with respect to cyanobacteria blooms. But is there anything from your perspective that needs to be done to, other than a registry, to start addressing this? Oh, somebody else has a question, so I'll take that in 1 second. But anything else that you can suggest from a systematic level that you think needs to be done to address it?
I think I've addressed the main issues. If the medical society was able to send out information to their members, most physicians in the state of New Hampshire are members. I think that would be helpful. Webinars put out by the DES, or having the movie The Lake Effect played on New Hampshire public television might be useful. I don't really know how you get the public more educated. It took a long time to convince people that they shouldn't smoke cigarettes, but the rate of smoking's gone way down, at least in this country. And it's still not believable to everybody. The state toxicologist for the state of Vermont didn't. She was berating my research over and over and over again, and eventually we were able to get a registry. But it takes a long time to educate people sometimes.
Yeah. I'll take is Peg, is that you that has a question?
It's actually Representative Erica Leon, who's sitting here with Peg. My question is, while you were talking about this, I also looked up some of the other ways to mitigate cyanobacteria blooms. And between that and what you were talking about, is it realistic to say that it tends to accumulate on the downwind side of standing bodies of water that are in the right conditions in order to propagate these blooms?
Under the right conditions, when they come to the surface, they will be blown to the downwind part of a water body. That's pretty typical.
And then with that, is that something where it might make sense to talk about the Department of Environmental Services to perhaps see if some of the created standing bodies of water may actually be increasing the risk in smaller pockets of cyanobacteria blooms, in ones that may be under the radar because we're focused only on larger water bodies?
Yeah, there are lots of smaller water bodies that have this problem. And, designing septic systems that don't leach into the water, having buffers around lakes. You can't do anything about global warming as of yet. People are trying, but it's not going to be solved overnight. And apparently the amount of phosphate that farmers in Vermont have stored over the years in the soil is so great it would take hundreds of years to get rid of it. But if you have buffers around water bodies and you have good septic systems and you keep people from putting fertilizers on their lawns, and all these things can help. Not putting down asphalt around water bodies can be helpful, because otherwise everything doesn't go into the ground, it goes into the lake. And the Charles River in Boston's had terrible problems with cyanobacterial blooms. And it's probably because there's too much asphalt and there's too many drains that just go directly into the river.
And it sounds like you're saying that either if it flows too quickly to a body of water, or if the body of water is more stagnant, that there's a higher risk. So would water management and wetlands management processes that would encourage more natural flow of water from one place to another and not artificially increase or decrease that flow, perhaps mitigate this risk as well?
I'm not an ecologist, but I think that makes sense.
Okay.
And I don't think that New Hampshire necessarily has done a great job of keeping their wetlands from being developed. I saw in Lebanon, New Hampshire, I saw a hotel put right on wetlands along 120 as you come into Hanover. And that was true marsh. And they got big bulldozers in there and they completely flattened the marsh and put a hotel on it.
I think these kinds of questions we should reserve for DES if we
Yes, I think
decide
so too.
among ourselves to have DES speak to us about that. I really would like to stay on the topic of the health effects associated with
Yes.
cyanobacteria blooms and ALS today, if that's possible. And so if there are any other questions about that in particular, and just reserve the questions about those types of things for DES would be great. I noticed that Representative Munns joined us. So Representative Munns or Dr. Timmons, do you have any questions for Dr. Stommel? I don't know
No,
how
I'm
much
fine.
you were.
I'm
Okay,
fine,
great.
thank
No
you.
questions. Thank you.
Sorry, I'm late.
Okay, great. So thank you so much for this presentation.
welcome.
It was great to have you come speak about this important subject. You're welcome to stay with us if you want to for the rest of our meeting, but thank you so much for doing that.
Okay, thank you. I'm gonna actually have other stuff to do. So
okay, great. Thank you so much.
Thank you for
You too.
inviting me.
Great. Next on the agenda was an update from DES and DHHS, and I think DES is not able to attend today. So Michelle, do you have anything from DHHS to update us with on?
Yes, I can share a little bit. So this is focused on the Merrimack cancer, the kidney cancer investigation update for you. So the department contracted with Dartmouth College, the state's academic partner for the phase 3 feasibility study. It was approved on December 20th of this past year, 2023, at the Governor and Executive Council meeting. The contract is expected to be completed by June 30th of 2025. The contract has a 2-year extension that is available in the event the timeline needs to be extended, but we're confident that we should meet the deadline of June 30th. Dartmouth College is well positioned to complete the study successfully, having operated and maintained the New Hampshire Cancer Registry since 1985, with an established and well-developed cancer data reporting and analysis mechanism. Dartmouth is the only National Cancer Institute designated comprehensive cancer center in New Hampshire, allowing for ample resources, including academic researchers. The phase 3 feasibility study will be based on New Hampshire's Cancer Concern Investigation Protocol, which includes convening both a community advisory board and a scientific advisory board. The team is now seeking recommendations for members to serve on either board. Feel free to reach out to me if anyone is interested, and I can certainly pass it along to the team. The community advisory board will represent the needs of the current and/or previous residents of the phase 3 feasibility study area of interest, and provide input into the feasibility study design, completion, and communication plan to the general public. The Scientific Advisory Board will provide expert input for the feasibility study design and completion. A steering committee, which includes staff from DHHS and Dartmouth, will provide general project oversight to ensure that work plan timelines and deliverable milestones are met. And a core team of staff have been meeting weekly to initiate the project. We did share this same update at the HB737 commission meeting last week. So they are aware of this, and I know there are folks that serve on both commissions that are interested in this this study. I also wanted to let you know that CDC ATSDR updated their PFAS information for clinicians, and it was just released yesterday. And so this document provides updated information for clinicians to consider when seeing patients who have concerns about PFAS exposure or possible health effects. I apologize because I have a little bit of a cough. This was last updated in 2019. This update incorporates evolving science and emphasis on the importance of shared decision-making process with patients and clinicians working together to address patients' concerns and reduce PFAS exposures. In order to support discussions with patients, the update provides information for clinicians on exposure sources and routes, health effects associated with PFAS exposures, and factors that impact the risk of health effects. Consideration for clinical evaluation and management, including information on the benefits and risk of PFAS blood testing, and considerations for certain populations, such as children and those that are pregnant or breastfeeding. And they include additional clinical resources and information to assist providers and patients. I will drop a link into the chat so people can access this on ATSDR's website. And we are currently reviewing this update. As I said, it just came out yesterday. So we're in the process of reviewing it, and we will update our New Hampshire PFAS guidance for clinicians as necessary. As you know, we largely used ATSDR's guidance for our guidance. So we will review this and update New Hampshire's guidance as needed. And that's what I have for my update, and I'll be happy to drop a link into the chat so people can access this new guidance from ATSDR.
Any questions for Ms. Roberge from the commission members? I should have said doctor, shouldn't I have? Should I have said doctor? Sorry. Okay.
Actually, I have
I'll
a
try to be
quick
very
question.
careful.
I have a question that's mostly
Sure.
related. With the links, can we get all those links from the chat emailed to everybody afterwards so that we can have access after the meeting ends? Because that's when I go back and follow up. And thank you for the update on that. It's great to know what's happening with the kidney cancer since we worked so hard to make sure this would go through in a good way.
Yes. And I know Representative Leon, you were a big help with that. So it's exciting that we're rolling it out now. So
that's good to hear. Thank you.
I'm happy to send them, or is someone going to be able to get them out of the chat?
I'm copying them. I'm taking them out right
Okay.
now and putting them
Perfect.
into the agenda, into the notes that I'm
And
taking.
I can, when New Hampshire updates our guidance for the PFAS, our PFAS guidance for New Hampshire clinicians, I'll let I'll provide an update so you all know that our guidance has been updated as well. It may take a little bit. I'm just going to put it out there because we are certainly very busy this legislative session. So we're kind of balancing a couple of things, but we will make that a priority and get that updated as well.
Any other questions from commission members? I have a question, actually. I noticed in the news that the insurance commissioner took a step to require insurance coverage for PFAS blood testing in New Hampshire. Are you familiar with that? And if not, don't worry about it. Maybe we can figure out a way to get somebody to speak to it.
That I'm not sure off the top of my head. I thought that was covered, but now I may be.
Well,
I don't
it's
want to
in part
misspeak.
covered, but
okay.
Yeah. And then just like this week, I saw maybe Senator Ricciardi knows about it.
I was just going to say that while we were here, the commissioner was texting me about a different matter and needs to speak with me Monday. So if you'd like, I'll ask him.
Sure, absolutely.
Okay.
Yeah, I saw it in
And
the
then I'll
news,
send
so
you
that
an
would be
email.
great.
Okay,
Excellent. Thank you so much.
you're welcome.
Any other questions for DHHS? We'll move on to two legislators will join us. I'd hope that two others would be here, but Chris, Representative Munns, anything, and Senator Ricciardi, anything from your end, and Representative Leon, anything that you want to bring to the commission attention that has to do with any of our the goals of this commission? Maybe start with Chris. Put you on the spot.
Thank you. I don't know if you already covered the subcommittee reports, but we did get a report from DES providing an update on what has happened and what is proposed to happen at the Coakley landfill. I haven't had a chance to look at it in great detail, but I think the opinion of those that have and is that it leaves something to be desired. It's the hope was that it would provide a picture of what the plan is going forward in terms of finally addressing the problems that have been happening at Coakley. A true remediation plan. And it appears that basically all that is in the report is, more testing, more monitoring, and no clear sense of what the overall approach is going to be, or what the overall solution is going to be. When I had spoken with DES about this late last year, I had said that after we had received the report that we'd like to have an opportunity for legislators in the area and commission members to meet with DES to kind of go through the report, and that also hopefully there'll be a public hearing. So I need to follow up with them on that. I've been a little bit preoccupied with the ramp up in legislative activities and haven't been able to do that, but that's the plan, is to follow up to have those two discussions and really try to get a sense of where we're going.
Great.
I think that's about all. The other thing was that I don't know if he's on the call, Representative Muse. I had reached out to him about collaborating with him on putting together the legislative update spreadsheet, and he shared that with me. And again, I've been preoccupied with my own stuff on my committee and some bills that I'm sponsoring, and haven't had a chance to work on that. And hopefully I can do a better job before the next meeting.
Do you want to just share that with me, and then we can use that to start off, and maybe have everybody pile on if they have
I think
ideas to add.
One of the things that David and I talked about was trying to make that kind of a Google Doc that
that'd be great.
we could collaboratively update. And what I can do is I can try to copy paste that. I can try to take the spreadsheet that he has and create that Google Doc and share it with him and with you.
Great. That'd be awesome. And then we'll just share it with the rest
Okay.
of the commission members. I know there's
Okay.
a lot of stuff going through, including there was a hearing on this commission yesterday. So we definitely don't want to lose sight of what's going on in the legislature if possible.
Okay.
For us outside. Anyone else, Representative Leon or Senator Ricciardi, do you have anything that you want to share in terms of legislative happenings?
I
The
think
only thing, I still don't have, the hearing has not been set for my bill on the PFOA remediation. I know that's partly the other commission, but it's of interest to this one as well. And when I know that hearing has been set, I will let everyone know.
Do you have a bill number for that?
Do you know on the top of my head? I don't know. But
Okay, I'll look. Yeah. And did I hear Representative Leon say
yes.
something?
Yes, we had the bill hearing so what number is it? House Bill 1171, extending this commission. We had that yesterday. I put in an amendment with both myself and Nancy Murphy to remove one of the charges of the commission that's now outdated. It's number 11, which is reviewing the results of stages 1, 2, and 3 of the pilot study recommended by the previous commission established in 2017. Because that is now out of date, and also amending the data sharing to be every 12 months instead of every 6 months, because Representative Murphy said that was a very important change. So we have an amendment in to do both of those. And my read of the room so far seems that those seem like some reasonable changes in the education done by this committee. Even just that itself is enough to keep this going, because that's a really, really valuable service. So we don't even get into discussing anything else, because that education that's happening is so good that I think that guarantees this to continue on.
Thanks for those comments. I wasn't able to attend, so it's good to hear that.
It was at the end of the therapeutic cannabis, so not a problem.
I see the hand up. Michelle, do you want to say something?
Yeah, I just wanted to let Representative Leon know. So there was a bill last session. It was HB 391, which had to do with the MOU that's tied to this commission. And there was some language put in there, and I apologize because I haven't been able to go back and look, but I know that there was some language that was put in that changed the reporting date to annual. And I know that it also tied it with HB 737. So I just want to make you aware of that. In case that it's already in there somewhere, we may just have to reference back to whatever the language was in HB 391 last session. So you may want to just reference that. And I apologize because I haven't had a chance to look back and trace it back, but I just wanted to make you aware of that.
I appreciate that. We were working with different versions, so I will make sure that any of those changes we know of as we're working through, because I know Representative Murphy had another section to change that may have already been done in 391. So I will follow up on that.
You may want to just track that. Yeah. Thank you.
Thank you.
Anything else on legislation? Senator Ricciardi?
Oh,
So she can't hear me.
I just got a text back. I can answer your question now. The commissioner said that bill has only been on the periphery of the department's focus. So he's going to check on it, and he wants to know how the department can be more helpful. So I think we're going to have to pursue that.
Okay, that's great, actually, because there are some concerns around that availability. So
Okay.
I know there's a bill passing through this session from Representative Murphy that has to do with that, expanding access to PFAS blood testing. So that's important. So maybe we could actually get that connection, have them stop through here in one of our next meetings and see if something, we'll chat it up, figure out how to do that. That's great.
Okay,
Thank you. Along those same lines, Michelle, there had been a question about the reports, the content of the reports. And I was thinking a little bit about some of that, and I wondered if the agencies were open to some suggestions on content.
You mean for this commission, SB85, the annual report?
The annual report.
Certainly happy to hear any suggestions. I mean, I know we've been doing it. We have a sort of a format that we've been working on. And I think we were trying to combine the work of 737 and here, or the report. There's something about that, and I apologize. I don't know it off the top of my head, but certainly happy to have a conversation on that. Yeah.
Okay. If you're still open to that, I think I would suggest, we are kind of running out of time for sure today, and would suggest that the commission members take that back under advisement and take a look at those reports and see if you make any suggestions about how to, whether the content, if there should be additional content. One of the things I was thinking about was there's so much stuff going on. Maybe it'd be helpful to put in a little bit more about, say, the trace programs or the biomonitoring program. I know it was referenced in the PEAS study in particular, and it might be good to have a little bit of an update in there on, say, the New Hampshire, the biomonitoring study a little bit more in detail.
Okay. Yeah, I know that there is a little blurb. I mean, what DES and DHHS kind of work together. We sort of have this format in place,
Yep.
and we do periodic updates. We work together and try to just submit one report that's sort of blessed through both sides. But yeah, certainly we can, happy to take feedback, and where we can provide more information, happy to do that. And as I said, I think it's now an annual report as opposed to every 6 months. So there might be where there's a little bit more time,
Yeah.
it might be a little bit more comprehensive
Yeah.
anyways, just by the nature of the time factor. So
Yeah. What I was thinking of too is if it could be tied into the content of some of the things that this commission has been dealing with. So, we're next meeting, this is kind of a good segue. Next meeting, we'll be hearing about radon. And as long as the schedule is, I have to check with you guys to make sure this is still right, but I have it on the calendar that next month is going to be a presentation on radon.
Yep.
And then in March, we'd be hearing from Gail Gettins on lead. And then in May, we would be hearing on the EJ and climate issue, science, the study that's been done, I think it was at UNA. And in there, so we have we have April open still, but those types of, hearing about radon, hearing about maybe the lead program in particular, sort of update, the things that we are interested in hearing, the cyanobacteria thing. If we could make that content kind of marry up in the reports, that would be great. Is what my thought was, just in general.
Sure. So I can confirm that Lynn Clement is prepared, or will be prepared, to present on the 16th of February. I asked her for a time frame. She figured about 20 minutes, and then obviously questions. Gail, yes, I can confirm Gail is for the 15th.
Great, of March, yep.
Yep, and we can give you an overview of the lead. I'm assuming it's like an overview of what the department is doing relative to radon and what the department is doing relative to lead. Okay.
Yeah.
And then I did notice that biomonitoring is on there. And I did reach out to Dr. Chatterjee on that, and yes, they can do it, but I didn't confirm a date because I wasn't sure what was available. So
April?
I can check with her. Is April or May available, or is there
May is taken.
May is taken. So April or June then maybe?
Yep.
Okay. Let me confirm with her, and I can email you and let you know. Okay.
Great. Thank you so much.
Sure.
And if I can interject, House Bill 391 did already extend it to annually, so we don't need to worry about it in this bill, and we can get into that more detail. So thank you for bringing that to my attention.
Thanks. I just lost my train of thought again. Shoot. There's one more thing I had to say. Darn, I forgot. Anyway, any other new business from members? Senator Ricciardi, did you offer to have one of the in-person locations in Bedford or someplace near you? I can't remember if that was, it'd be, so what I'm getting at is February and March. It'd be nice to have locations locked down for the in-person. And I don't know if DHS.
I could look into it, but I don't, we didn't, no, I did not. I could see.
Oh, okay. I'm sorry. You weren't there. That's right.
Yeah, no, but if you'd like me to look, I can try.
That would be great. So what we generally are trying to do is move around to different towns. And we had also asked about DHHS and DES separately, seeing if they could sponsor the in-person location. So what we do is, as Peg so nicely did this month and Chris did the month before, was we just get a location in the library. And it's usually, so the next one is February 16th. The next one after that is March 15th, 3 to 5 p.m. and try to have a room in the library that's accessible to the public, and then just have AV equipment in the room in case more than a couple of people show up to be in the in-person location, and then just have somebody there with a connection via Zoom.
Okay, so I will ask, but when you say someone there via Zoom, I'm not technical, so I hope you're not asking me to do that. That would not be something in my wheelhouse.
Yeah, it's just connecting, usually just connecting a, the cord up to your computer is what we do.
Well,
so it's not really
ask
that.
if they have availability. I will say that library is like pre-booked out.
Oh, okay.
Like it's a very, very busy place. I will inquire, but I would say it's probably not. I'm going to say my guess would be it wouldn't be available, but I'll ask.
Okay, great. So either for February, March, and if anyone else can, and if DHHS or DES could as well, that would be helpful. If not, we'll go back to Portsmouth or Rye or Hampton or maybe Merrimack again. So we'll see what we can do. I think Nancy offered to have it again in Merrimack.
Okay. And my other issue would be with my schedule. I have like 3 meetings on Fridays and I'm trying to not miss anything. It's hard for me to commit. You know what I'm saying? I don't want to say, and then I can't be at the meeting.
Yeah. Okay.
Yeah.
Okay. If it doesn't work, it doesn't work. That's fine. Okay, great.
All right.
Anything else from anyone? Did you want to say something, Representative Munns? I see you turned the camera back on.
No, I just wanted everybody to see my smiling face.
Well, thank you. I think that's it. And thank you for all your great questions. I think that was a great presentation by Dr. Stommel. It was really informative. And what do people think about a DES presentation on cyanobacteria to this commission as well? So to get their perspective, yeah, we have some questions. Okay, so I'll bring that to Karen. I assume that would be Karen's area of interest to set that up with us. Yeah, because she did talk about that. I can't remember what she said, but I'll connect with Karen and see if we can get them to come maybe in one of our slots to come and speak about that to us. So thank you so much, everyone, for your attention, and see you soon. Take care in the snow.
Thank you. Happy weekend.
Bye-bye.